Hello, everyone. I'm going to give kind of two talks today. I'm going to first talk about clinical genomics, and then I'm going to take a little bit of a right turn into kind of the hybrid cloud area and a lot of the work that we've been doing on the R&D side, and you're going to find that there's a lot of parallels and overlaps with some of the talks this morning. In particular, I want to call out a couple of things that Ari mentioned. Scientists build their own clusters. They build them under their desktops. They can buy them. They can buy pretty good clusters for pretty cheap, and they can do this. And this has been a motivator for us because we can't get scientists to stop doing this. The other part to that is we're trying to figure out how to deal with that and how to build infrastructures that take account for that. And I would point out that in the first talk from IDC, the departmental computers are outstripping HPC right now. And we're not seeing that trend going away, and so we're trying to deal with that. And I'll get to that in the second half of this talk. So a lot of acknowledgments, can't take the time to do them all because it would literally take me all day. I'm talking about projects that have been going on for 5 years that involve a lot of people. I should talk out Dr. Kirk Wilhelmsen. He's a partner of mine. We're colleagues. He's the domain scientist. I'm the computer scientist. He's the one that's really driving a lot of this work, and I help him use computers. So we have, okay, that's better. We have kind of two areas that we do research in and that we support. One is on the basic research side of genomics. We have a very large NIDA-funded project to look at the correlates of genomics with addiction. And for this, we've been sequencing a cohort of 5,500 patients. We're probably about 3,000 to 4,000 right now that we've already sequenced. We're also doing a lot of work on the GWAS side on Alzheimer's. Then on the clinical side, we've been funded by NIH on a couple of different projects. One is called NC Genes. This is a project to actually look at the ethical, legal, and social aspects of bringing genomic sequencing to clinical care. And this project is slated to sequence 2,000 people in a clinical setting. I think about 700 to 1,000 have been done at this time. There's a related project called NC Nexus, which is sequencing newborns and looking at the clinical application of their sequencing. And then ClinVar, which is an NIH-funded project to look at the clinical adjudication of the variants and their clinical relevancy, which is a very large consortium that UNC is part of. I should also say that those are the projects that RINC is focused on. Some of the stuff that I'm talking about today, the workflows in particular, are being used by the UNC High Throughput Sequencing Facility. And so these impact the ENCODE project, the Comprehensive Cancer Genome Atlas project that UNC is doing. And so in total, we've actually run a very large number of samples, much more than the 5,000 listed here across all the different projects at UNC that are using their high throughput sequencing core. This is a high-level view of our pipeline. This is a clinician's view of this pipeline. This is actually a clinician's rendering of our IT version of this pipeline. And so you can see more of a clinical aspect. You have the clinical researcher operations. They draw blood. It goes to a wet lab. They do high throughput sequencing. The HPC part of this is a very teeny pipeline down at the bottom. They don't think of that as a big part of this. We put all that data into a database we call VARDB, where people can analyze it, integrate it with other data sets. Then that goes through a binning process to pull out the variants that are actually relevant for clinical care. And then it goes through a whole process of review by the clinical side. And so that's how our clinicians view this kind of pipeline. Those 5,500 patients I mentioned is that little teeny dotted box called research process, which the clinicians view as something else, but it's a big part of what we do. Again, the clinical side, this is the output of this program, and I should mention this is a in-production system. It's been running for a couple of years now. At the output of this, a patient comes in for a study, they get sequenced, they go through that whole process. The output of this is a decision support tool that the clinician, the review team can look at. Each line is a variant in a person's genome that has some relevancy to a disease that they are presenting with. Now I'll show you more specifically. This is a particular variant. This is a missense variant, meaning it will change the protein coding structure, so it could possibly have a bad impact on that person. This particular genome, if you see over on the left, VUS, that means it's a variance of unknown significance. We actually don't know what it does, even though we think that it does something bad. And you can see notes from the clinician when they reviewed this case, looking at what they think the literature says about the impact of this case on that person. This is one of the highlights of our work. About a year ago, a patient was referred to the study. The patient had been living with paraplegia that was diagnosed a long time ago. It was a false diagnosis. She's been wheelchair bound for 15 years. She was referred to the study, had her genome analyzed. An incidental finding realized that she had a condition called dopa-responsive dystonia. Now that is a treatable condition. There's actually a pill you can take for that. She's now walking 2 weeks after she came in and had the study done. So we were able to essentially cure her of that condition by doing this kind of sequencing. Not every case is that dramatic, but it's having an impact. When we talk to the clinicians, their estimate, ballpark, 40% of the patients now have answers for something that they didn't have before they went into this. It may be lifestyle things, it may be familiar things, it may be just having a better diagnosis. But these kinds of things are now in practice. They are having an impact in the world. And I should mention this was put into a Natural Journal as a paper, but it was also put into the prestigious Cosmopolitan Journal. Research side, we are doing basic research looking into the correlations between genotypes and phenotypes. In this case, and I'm not going to do much on this kind of talk, but this is just some early leading results showing a genotype association with lifetime and effect cocaine use. And you can see there's an area that's been highlighted with a little circle, and that's showing some possibilities of genotype relationships with that condition that will be studied more in depth. Probably the unique thing, and there's a lot of groups that are doing this kind of sequencing nowadays, and there's nothing usually unique about what we're doing other than we're doing it. What is probably unique about the way that UNC is doing it is when you have a dollop of money and you're going to do sequencing, you have a choice. Can you sequence a lot of people and have error in that sequencing, or should you sequence few people and have less error in that? And that's a money trade-off. And you can see you get kind of a curve. It's a made-up curve. But UNC, our sequencing on the basic research side is on the far left end of that scale. So we have a different strategy. Not everyone takes that strategy. This is a sample of coverage that we do across a number of patients. And you can see that this coverage across patients is somewhere in the 5 to 10 range. This is very light coverage. Most people do 30 to 50 or even 70x coverage. And coverage means the number of reads of a genome you do at every particular location. The more coverage, the less error you have. The less coverage, the more you have to rely on other approaches to deal with that. But we went for large sample sizes, low coverage. That greatly impacted the nature of the wet lab work, because when you go for low coverage, you get high variability. And so we have to do extra wet lab processing to figure out how to get the correct stoichiometry before we do the sequencing. I'm not going to talk much about that, but I did want to mention it. But it also impacts how we analyze this data. So one of the big things that we do that not everyone relies upon as much is doing imputations. And so the basic gist of this, if you look on the left-hand side, you have a patient, and they have in one location 5 Gs, in another location they have 3 Gs, an N, and an A. So in that case, you don't really know, is their mom giving the A and their dad a G? Are their mom and dads all giving A's and there's one unknown and one G? We really don't know. And so the way to deal with that is kind of traditional statistics. You look at other people. And so if your person has G's and you find that everyone else has G's in the red location, then you can infer that your patient has G in that other location. And so this is kind of traditional hidden Markov modeling, but it's very, very computationally intensive. And it's something we don't do very often, but it does well up the pipeline, the processing, and this is probably our biggest HPC part of our analysis right now. But it does work in practice. So if you look at this is a chart showing the correctness of the callings when you do imputation. So if we sequence someone, we do imputation, and then we resequence them using an exome chip, which is a different way of doing it that has a higher accuracy. And then we look at the correlation between these, we can see how well the imputation does. The green is the imputation that we do, and until you get down to very low coverage, you have very high accuracy in actually making those calls successful. And that means we can actually do a 3x increase in the number of samples we do to get the same accuracy. And again, I just mentioned this as one of the ways that HPC has come into the process of doing genomics at scale, because I'm sure most of you know about the genomic pipelines that run BAM and that generate BAM files and FASTQ files. But to those, these are pictures of two of the pipelines we run. One's the RNA-seq related to the Comprehensive Cancer Genome Atlas, and one is whole genome sequencing, which is one that we do for our sequencing of the addiction patients. We had 4 versions of this, a single machine one that we got 2010 or 11, a cluster-based one that I wrote back in 2011, and then we've had a better programmer rewrite those twice to run on multiple clusters and grid. And that system is called MapSeq. It's currently our current solution. It runs in production all the time. It's a Condor-based system. This is essentially the schematic for MapSeq. The top left, we have the MapSeq system itself, really a database that keeps state on all the processing that's being done, all the pipelines that are being done. And remember, there are hundreds of pipelines being run every day on this. A bunch of Apache technology to let people submit jobs and to keep the relational mapping between the software and the database in line. So a person can submit a request RESTful to this. The system will recognize that. They'll grab the right pipeline. They'll push it off into Condor. Condor then is aware of the resources that we have in-house, but we also have a system called Gate that monitors Condor itself and sees when we need to add more resources to the Condor pool, and so can create glide-ins or kill glide-ins as we need to. And we do this because we have a lot of clusters that we can get access to locally. We don't own the clusters, and so we have to use them opportunistically. Our main cluster is one called Curry. We actually have 34 nodes that are dedicated to us in a patron kind of queue model, but we spill over a lot of times because sometimes we fill up those queues and we need to spill over. So we have 2 other clusters at RINC and ITS that we use. And I could also point out that some of you may remember from many years ago, TopCell was one that was on the top 100 list. It's now was mothballed. We brought it back as an educational system. We run Hadoop on it for classes, but we've also been able to pull that system in through these Gate plugins, and that's actually been run on a couple of times where we bumped up against limits of what we have in the queues for those systems. So MapSeq has been, it's custom written, it's open source, it's available on GitHub if you want to look at it. It's been a great time savings and efficiency saver for us, mostly because it has allowed us to access multiple clusters optimistically without having to go out onto something like OSG or to get time on some of the big machines. It's pretty good software, minimal intervention once we get it set up. The pipelines are modular. We have 40 pipelines we've written. 20 of them are in production use right now. They can be tailored, modified. And so it's a good solution we built up, and as I said, it's in production. When we look at that, though, we've had lots of challenges that we are trying to address in the future. They are complex. Not many people can run these kinds of systems, frankly. We have risk in that there's only a few people that know how to do this. We can't transport these to other groups very readily. And we don't really have a true cloud model. We do have access to machines, but not in a cloud-like way. Then we start looking down the road, we see bigger challenges with the analysis that we have to start doing. So how do we bridge the islands of data where data is stored at different locations? It's hard to move because of cost, security, regulatory, privacy, and size issues. These are huge problems for us right now. that's causing us to inefficiently use resources between our collaborators, and it's dictating how collaborations are being done right now. So this is something that we're starting to look at. Part of this is using the iRADS technology. This, if you don't know about iRADS, iRADS.org on your browser. But essentially, this is a data grid that lets us virtualize where our data is located. So you're looking at a snapshot of a bunch of our NC gene data. You can see each file in there. There's metadata on the files. We can share it and things like that. You don't particularly know whether that's on tape or on a disk drive right now because iRADS virtualizes that for us. So that's one of the solutions that we're moving towards more aggressively on the R&D side. The other thing is that we've been doing a lot of R&D really to understand and apply hybrid cloud models into this realm to try to address some of these challenges. We've been in particular using one, ExoGenie, with Rincy does with Duke University as part of the Genie program at NSF. And this is essentially a network infrastructure as a service. So you can ask for VMs and you can ask for the networking that connects those VMs as a layer 2 protocol. And this is kind of nice. It's essentially a cloud of clouds. And so we're looking at how to use that in the life sciences. The nice part about this is conceptually, we can look at this as a collaborative virtual infrastructure. So I, who sit above that blue line, can say, okay, I need a slice, which is machines and networking between them, virtual private networks between them, and I need to do cloud bursting. So I want to grab a bunch of VMs and do that. On the right-hand side, though, this is a more common case, becoming more common. I have a group of collaborators, and I need resources for them, and I need them to be in a virtual space that I control or someone controls and not out in the Wild West. And that's probably the one that we're focusing on a bit more than the other. And then I want to make sure that the IT is there so I don't have to make 10,000 phone calls to orchestrate this, which is what I typically have to do today. Part of this is coupling this with iRADS, because when I have that slice now, that virtual space that I can operate in, I can reach out of that space not on a file name basis or directory basis, but on metadata basis. So I would like to be able to say, I need data from lab X, and I'm user Y, and I'm running project Z, and deal with things at that logical level and not at the IT level that a lot of scientists are not comfortable with. So our first real foray into this, we demoed this last year at Supercomputer 14, was to push our genomic workflows out into the cloud, into this kind of cloud. And there are really two models. One is taking our existing workflows, which run through MapSeq, which already go out to cluster 1 and 2, and actually spinning up VMs out on this cloud infrastructure. And we've been able to do that. We wrote a gate plugin that lets us now have MapSeq push things out into a cloud. That's a kind of more complex solution. We actually spent more time looking at what to me is a simpler solution, which is we have hundreds and thousands of workflows that we run. We don't really want to break up the workflows by their pieces. We want to deal with the workflows as ensembles. So we wrap up the entire workflow in a VM. We run the entire thing on one single VM, but we look at how many workflows we can push out into a cloud-based infrastructure. Now, the workflows themselves, yes, they run a lot longer when you put them on a single VM, but I can cloud burst the entire thing and deal with things that way. And so I have a simpler model, everything's wrapped up and clean. Again, this is R&D, we're exploring this as possible solutions. A good part of what allowed us to do this is a collaboration with the Pegasus team. Pegasus is a workflow engine. We've engineered with their group the ability to start a workflow using a few nodes. Pegasus can then predict how many nodes we're going to need, ask the infrastructure, the network infrastructure as a service, for new resources, so spin up new VMs in anticipation of jobs to be done, execute those jobs, and then tear it down when they're done. So this is my last slide, and I have 3 minutes to explain it because it's very ugly. This is our latest project, and really what we're looking at now is the problem of how we build these kind of collaborations in a way that's secure, in a way that's virtual, in a way that builds a virtual space where different people can collaborate within. And the gist of this is that we constantly try to build collaborations with groups that inherently do not want to share data, but that need to share data. And if you know where we're located, we're at UNC, and our partners down the road are Duke. And Duke and UNC compete heavily on the medical side, but they also need to share data on the medical side. So Duke doesn't really want to give UNC outcome data on how many cancer patients died, but they do want to share information about those patients. So we're taking those concepts I was just showing you and looking at how we build distributed virtual spaces that are secure and that we can do computations within. Now, one of the things we're doing right now is a project called RADI. It's an NSF-funded project to look at how do you describe such a collaboration. You know, there are machines you need, there are data flows that you need, there's processing capabilities you need, and that typically is described by IT people in long conversations with other people. We want to bring that up to a level where we can describe that more like a data flow, and then have the system instantiate all the resources we need for that. And so that's what we're working on right now. We hope to have some demos, so this is a shameless plug for the networking team. If you come to SC15 and come to the RINC booth, hopefully we can be demoing some of this. The other part to this that we're looking at now is how to do privacy-preserving calculations in a distributed way on these hybrid cloud infrastructures. We did our first test a couple weeks ago. I successfully took a GWAS analysis that normally takes about 1.4 seconds and made it take 14 minutes. That's not really a success, but these kinds of calculations are very, very computationally intensive if you want to secure privacy. And we see that as one of the directions that we're going to have to start understanding how to deal with in the HPC world, how we can push calculations off, keep them secure, and do distributed calculations on these kinds of infrastructures. And so that's my talk. Thank you.